Well, this begins the second of this sensory physiology series of lectures, and it is the final one for sensory physiology. In this presentation, we will consider the eye and vision. Obviously, this is an introductory level discussion of vision, and it's quite more sophisticated than this. However, this will be sufficient to get us started in our gaining appreciation and understanding of the special sensory function of vision. So, as with the vestibulocochlear, we should point out, I guess, that we would call these special somatic afferent systems. So the sense of taste, as we described it, was the special visceral afferent, and smell was special visceral afferent. With these, we would consider them special somatic afferents. So cranial nerve 8 and cranial nerve 2 would be special somatic afferent for their sensory components. So the light enters the eye, as we know, and it's focused on the retina by the lens. So this is the basic arrangement, and then the photoreceptors will transduce the light energy into an electrical signal, a biological property, as we know, that will be processed through the neural pathways. So we begin with this kind of an odd view of the orbit. We see the bony skull here on the right-hand side of an individual, and there was the boundaries of this with the eyelids attached laterally with this ligamentous kind of a structure, and then of course medially we see the ducts from the opening between the eyelids into the tear duct as we know it, the nasolacrimal duct that drains the tears into the nasal cavity. So this originates with the lacrimal gland in the upper outer or the superolateral position within the orbit, and we talked about the neural innervation of this. Sorry, that was maybe another course. Just crossing my anatomy course with this one here. Anyway, there is obviously a sympathetic and parasympathetic innervation to the lacrimal gland to regulate the tear production for the eye. As well, their cranial nerve innervation to the eyes themselves, the extraocular muscles for the control of the position of the eye within the socket. And then there is, of course, a flocculent fatty tissue, adipose tissue, that surrounds the orbit within the orbital cavity as a cushion to protect. And then, of course, the strength is coming from the bony boundaries. So what we are focusing on here predominantly is, of course, the anterior surface of the orbit itself. The eyeball has the white outer connective tissue. We know that there's conjunctiva covering it here. But anyway, deep to that would be the sclera. And then as this continues over the central midline anterior surface, the type of epithelium and connective tissue changes such that it is translucent. It allows the pass of light. And this is the iris. Then we can, through this we can see the iris, and the pupil is the hole in the center of the iris. So let's move on. So that translucent central circular portion that I mentioned is the cornea, as hopefully you were filling in. And through the cornea, from that anterior view, we can see the iris. So here we have then a sagittal cut down through the center of the orbit. It isn't exactly perfect like that. It's somewhat oblique. As we'll see, the optic nerve is actually off to the side a bit. But anyway, this shows us through a longitudinal section, although oblique, of the eye, the different anatomical structures that would be involved in the pathway of the light, and in the next slide we'll look at a bit more of the detail of that, and then we'll look at the retina as if from an ophthalmoscope. So zooming in first on this panel A, the sagittal section of the eye. First, let's just remind ourselves of the eyelids that we had talked about, and then there is a mucosal membrane, the conjunctiva, as you know, that overlies the white, tough connective tissue capsule of the eye called the sclera. And the sclera is very dense and thick, obviously, to contain the eye, and therefore there is an intraocular pressure, so it is a closed chamber. But there are two weak spots. One is posterior, where the optic nerve connects, and that area internally is referred to as the optic disc. There's no retina there, so it's called the blind spot. And then the other area where it is altered and weakened, you could consider it that way, is at the cornea. And of course it's altered such that the connective tissue is oriented in such a very refined way, very parallel to each other, to allow have a translucent property, to allow light to pass through it, without changing the pathway of the light waves very much. So then what we recall that, and the fluid dynamics is very important in here, as you know. So we won't get into the physiology of that at this time, but the canal of Schlemm is related to that, as you may know, in regulating the aqueous humor that is found in the portion of the orbit between the lens and the cornea. And you know that the iris separates that anatomical subcompartment of the orbit into what's called the anterior chamber between the cornea and the iris. Excuse me. And then through the pupil, the central circular opening through the iris, we have the continuity into the posterior chamber. And the fluid pressure in here has to be regulated, and you know that can be measured, and it changes in certain disease states. So anyway, the fluid dynamics is as important here as in the fluid dynamics for the vestibular apparatus and the cochlear apparatus, as we have described. So each of these fluid compartments we've been considering are minor fluid compartments for the total body fluid, the extracellular fluid compartment. So we've seen the cerebrospinal fluid as one site, and other than interstitial and plasma, we've seen the cerebrospinal fluid, and then the endolymph and the perilymph, and then here the aqueous humor, and then of course, although more gelatinous, the vitreous chamber containing the vitreous humor still is extracellular fluid in here that is exchanged continuously over time. It might be a slow rate or a low rate or a high rate, but still it is changed continuously over time. The lens, as you should know, is suspended in space by the ciliary bodies here that contain the ciliary muscles, and then the zonules surrounding the retina. The lens have these zonular fibers that attach to the periphery of the lens. And obviously this is a dynamic structure for controlling the length and the shape of the lens. And then we'll consider the light path through here as we proceed. The vitreous chamber is lined at its periphery for the most part by the retina, and the retina is a layered structure with the photoreceptors in it, as we will look at in a little bit greater detail. And the distribution of the cell types in those layers will differ throughout. So you're going to here close to the lens, you're going to have mostly just the monochromatic types of visual perceptions, or sensory detection capabilities, and then more and more of the color and higher resolution. And of course, as you know, the highest resolution at the fovea, that is a little depression, and it's a depression because we don't have anything between the vitreous humor and the photoreceptor cell layers, and a few of the other cell layers, so that and it's the highest density of the cones, the color receptors, so we get a very high resolution and color, multi-chromatic sensory structure here, the fovea. So when you're really straining to see something, this is what you're using. And it's positioned a bit laterally to the optic disc. And of course the vascular bundle, the artery and vein, the central retinal artery and vein that are a branch of the ophthalmic artery from the internal carotid artery that we'll see in a moment as we proceed, comes in and is distributed to the retina. Extremely critical. Any compromise to this blood flow and the retina is damaged, typically permanently, irreversibly. And we'll talk about other layers that are critical here as well. They're showing it, but they don't really have the layer labeled between the retina and the sclera, and that is a pigmented epithelial layer that will absorb light and prevent it from having a backscatter. And that, as you know, is present in humans, but not in all organisms. So with the ophthalmoscope, you can look through the pupil. Typically, as you know, they'll apply a substance that will allow pupillary dilation, so they can get a maximal view in here, and they can the clinician would look in and see this structure. Mostly what they're doing is they're looking for any anatomical abnormalities in the surface of the retina, like a retinal detachment or something like that. Excuse me. But in general, I'm sorry about that. This is elm season and my allergies are particularly bad, so if that continues, you may hear me just click off the microphone and then come back. So if you hear a silence, just wait a moment and I'll return. Okay, so we can see from the ophthalmoscope the blind spot, the optic disc. There are no photoreceptor cells there, and this is where the central retinal artery and veins are emerging into the retina, onto the retina, we should say, right, because it's on the surface between the vitreous humor and the axonal layer of the ganglion cells, as we'll see. And then positioned laterally from there, you can see the convergence of the small arterioles, even down to the capillary bed almost, at the periphery of the fovea, and this produces a spot-like structure, the macula. So macula just means spot. And as you may know, that there can be alterations or damage to the structure of the vasculature to the eye in different conditions. There can be localized hemorrhages or blockages or things of that sort, and it's very important to have this type of examination performed. So the neural pathways for vision and the pupillary reflex will be considered in the next couple of slides. So here we're looking at a dorsal view, a top-down view, as if this was an MRI, high-resolution MRI of an adult head. So we see the right eye and the left eye. Because it's top-down, we know it's a dorsal view, we know, and we can tell we're looking at the bridge of the nose here, so it's from above. This is the right and this is the left. And we can make out some of the portions of the cranial bone that are creating the cranial cavity and contributing to the bony orbit. And we see the flocculent fatty materials filling the orbit. orbital cavity, and extraocular muscles attaching to the orbit itself, and that would be the sclera that we're seeing there. And then from the optic disc, the optic nerves pass into the cranial cavity. And just in this area is the hypothalamus, where it would, and this is a section through the hypothalamus with the pituitary stalk. It would be attached there. And then this is a portion of the midbrain. I don't know if you recognize the substantia nigra here, but that's what that would be. We talked about that and this is the cerebral aqueduct going through the midbrain. So we want to recognize those portions. And then this is where the optic nerves from the right and the left first come together, and we're going to have crossing of fibers. There's always a decussation, as we are aware of in the sensory motor systems. So this is the optic chiasm, or crossing. And then the central nervous system portion of this is, although this is all CNS, but that portion centrally located to the optic chiasm is now referred to as the optic tract. And we'll follow the final destination of that in just a moment. We would have to be at a slightly more dorsal level to see the destination, final destination of the optic tract. It ends in the thalamus, so that would have to be above the midbrain here, and above the hypothalamus, correct? Dorsal to it. So first, let's just orient to a couple of nearby structures. Here we see the internal carotid artery. So the first branch of the internal carotid artery, upon emerging into the cranial cavity, is the ophthalmic artery, a branch of which becomes that central retinal artery. And then, of course, the venous drainage is a little more complex and sophisticated, but ends up in a dural venous sinus in this area here, the ophthalmic vein. And then nearby, we see the temporal lobe and some of the lateral ventricle, and I'm just wondering if you recognize this is the hippocampus on either side, and the amygdala just in front of it from our discussions in the earlier presentations on the nervous system. So anyway, just to kind of keep ourselves in mind here, so that earlier horizontal section was down through this level here. Actually, it was probably, I guess they were idealizing it coming through kind of like this. There's no real way of recreating it here, actually, from this illustration. But anyway, they had it going like this, and then you had some of the hypothalamus, and then it actually went through the midbrain here into the cerebellum, and you see the temporal lobe over there, right? So the hippocampus would be here, and then the amygdala sitting in front of it, and then here's the pituitary stalk with that infundibular recess of the third ventricle that we oriented to. So just to kind of keep the anatomy of the structures in mind, and that these are somewhat idealized illustrations or cartoons of these structures. But let's follow it now for the neural pathway for vision from this lateral view with a midline, a mid-sagittal section through the brain and the brainstem. So again, we see the third ventricle here joined by the lateral ventricles with the cerebral aqueduct into the fourth ventricle in that area there. And then this is the thalamus and the hypothalamus. So from previous lectures on these topics, that much anatomy you should recall. So we are seeing after the optic chiasm, which is just in front of the hypothalamus, just anterior to the hypothalamus, the lower portion, the ventral portion of the hypothalamus, obviously. But anyway, from there, the optic tract is going to pass on either side of the hypothalamus up to the thalamus and form the synapse between the first order sensory neuron and the second order sensory neuron in the lateral geniculate. And then from the lateral geniculate, there will be what's called the geniculostriate projection. There are stria, little strips of gray matter that can actually be seen even at the gross level in the primary visual cortex of the occipital lobe, and therefore this is called the geniculostriate pathway, or it is a thalamocortical pathway as well, the general term that we've used for any thalamocortical sensory projection up to this point. There are other projections also to the superior colliculus to help coordinate eye movement and the movements from the colliculus, superior colliculus. There will be pathways ultimately that will get like to the cervical spinal cord to control the head, neck, and shoulder muscles, some of the upper trunk muscles, sorry, to orient the head with the movement of the eyes. So there are other collateral pathways, and we'll see a few that get into the midbrain, even for the pupillary reflex in the next slide, I think. So this at least gets us through the pathway to the primary visual cortex, with a bit of a reminder about some of the neuroanatomy. So here we are considering in panel C the collateral pathways that leave the thalamus to go to the midbrain, and this is specifically related to the pupillary reflex, not for the superior colliculus that I had mentioned before. So obviously you want to coordinate all of this. I just wanted to say that for audition, for the sense of hearing, the medial geniculate and the inferior colliculus are involved, and it's a similar type of thing. You orient your head and neck by projections from the inferior colliculus down to lower centers, ultimately to the cervical spinal cord, to orient your head and neck to a sound, right? So sight and sound are going to have these. So one way to remember these is the lateral geniculate and the superior colliculus. That's light and sight, and then the medial geniculate and the inferior colliculus, the MI, like me, when you're warming up your vocal cords for singing, right? So that's obviously the audition. So that's one way to remember those. But it's important in sensory physiology to be thinking of these more complex collateralized circuits in their relationship to the complete response of an organism to the environment. So think of an alerting sound to your left. Your sense of hearing has to tell you that it's on the left-hand side, as we talked about, and then you move the eyes and you move the head to the sound, and it all gets coordinated. And then we'll proceed from here to talk about the autonomic nervous system in the next lecture, lecture 12. If it's a dinosaur about ready to chase you, then you got to have some system from all the visual information and auditory information you get to turn on your autonomic nervous system, your sympathetic drive, and your somatic motor system to run and get the heck out of there. So anyway, let's continue with this for the pupillary reflex. First let's consider the visual fields. They're not shown so definitively here, but we want to split them into a right visual field and a left visual field, again presuming that this is a dorsal view similar to the one that we saw originally. We're looking from top down, so this would be right and this would be left. So left visual field is coming in this way and getting the right retina there in the right eye, and then coming in here and getting the right retina in the left eye. And what we can do is when we follow that, so that's the left visual field, but the right half of the retina in each of the eyes, we follow those, so the right retina of the left eye crosses, decussates in the chiasm, and there it joins the undecussated, the ipsilateral, so it'll stay on the same side, that's called ipsilateral. And this is on the other side called contralateral. So the right retina contralateral will join the ipsilateral right retinal fibers in the optic tract. And then that will synapse in the lateral geniculate and you get the projection to the visual cortex, primary visual cortex, as we had described, through the geniculostriate pathway. But there's also collaterals to innervate other neurons here that will have projections to the midbrain. In this case, specifically to innervate the preganglionic parasympathetic neurons for cranial nerve 3 that will control pupillary constriction. So this is, remember, cranial nerves 3, 7, 9, and 10 have the autonomic control. Autonomic components, the general visceral efferent component. And then, obviously, it's the same for the right field of view, and we are then impacting the left retina of each orbit, and left stays on left, left eyeball stays on left, right eyeball, left retina, but remember this is the right visual field hitting the left retina, crosses. So you got to say those a few times and illustrate it or write it down to have it stick. So the right visual field impacts the left retina in each orbit, and the right visual field is what is transmitted to the contralateral primary cortex. So this is different than the other sensory distributions. The sensory input from the right body goes to the left cortex. So that's the sensory receptive field. So the right visual fields end up on the left primary visual cortex, but they impinge, they impact, and the neural signal originates on the left half of the retina for each orbit, and all end up on the left side. So practice that a bit so that you get a better feel for it. Clinically, it's very important, obviously. So the pupil then, the light enters the eye through the pupil, as we saw. The size of the pupil modulates light, and therefore will determine the impact on photoreceptor populations, where they're positioned, where the majority of the light hits the retina, and how much scattering you have. So if you're going to look far away, do you need the pupil more dilated to bring in more light? Or do you need to close the retina to pull in just a little bit of light to see far away? So that's what we want to deal with. That's what you should be thinking about right now. And then the shape of the lens focuses the light. So correspondingly, you need to change the the shape of the lens to either focus on distant structures or close structures. So we'll work our way through this. The pupillary reflex is a standard part of a neurological exam. So it's not only going to respond to the effort to look at distant or close structures or things in the environment, the presence of light or the intensity of light will also affect whether the pupil is going to be opened or closed. So if you're for night vision, are you going to open the pupil to let more light in with the hopes of increasing sensitivity, or are you going to constrict the pupil at night such that you try to see better? I say it that way because you don't. You want to open up to let more light in. But anyway, let's move on. So when we always see it, the clinicians, they'll flash the light in the retina and watch the pupil, right? And you should be able to see the pupillary constriction in response to it. So the refraction of light, we understand this and remember some of this from physics. So in a biconcave lens, you're going to get diffusion or divergence of the light rays or scattering of the light rays on the output side of the lens. So if you have parallel lights coming in, they're going to bend when they first enter the lens on the convex surface, concave surface, going in like a cave, right? So concave. And then again it will be bent so that they are farther away from each other. The distance between the two parallel beams of light will increase on the output side of the biconcave lens. So this is presented only to remind us that the opposite of that, a biconvex lens, is going to converge the parallel light coming in at some point remote from the, or distant from the center of the biconvex lens. So you see this line here is supposed to be positioned right down to the center of the biconvex lens, and that point where the light rays cross each other is called the focal point, and the distance from the center of the convex lens, biconvex, to that focal point is, of course, the focal length. So the focal length of the lens is the distance from the center of the lens to the focal point. So obviously the anatomical lens in the eye is more like a biconvex lens, or just convex lens, than any other type. So it is going to generate a convergence of the light waves coming in, the photons, at different frequencies, as you know, different wavelengths, and there will be a focal length for the any one particular shape of the lens, and that shape of the lens can change. The fluidity or flexibility of that lens, so that it can change shape, alters, it actually decreases with age. And I was surprised to see in the textbook that they mentioned that from the age of 10 on, this ability begins decreasing. So, and then when you reach your 40s or 50s, of course, as with all connective tissue, and this is a connective tissue structure, connective tissue changes in its flexibility, and we actually lose flexibility, so it'll always stay flatter, and of course that's for distant vision. So as we age, we lose our close vision, and this loss of the process that we'll be discussing, called accommodation, has a clinical term for it, and it's presbyopia. Presbyopia, as you can find in the textbook. So let's consider the normal convergence and accommodation for vision, and then see what's going on, why people would need glasses based upon anatomical inaccuracies, if you will, within this physical structure. And then talk about the changes. Well, we mentioned the changes with aging. We don't have any slides for that. But we'll mention the clinical terminology for common visual problems. So again, we're considering the focal length, and this is dependent upon the shape of the lens. Not just that it is biconvex, but the angle of the arc angle of the convexity, as you know, or in other words, the length, the overall length of it, and how flat it is, to put it in general terms. So a lens flattened will allow more distant light to be focused at the focal point with the same focal length. And they always show it going to the one spot, you know, and which is just the way we want it to work for the fovea. But remember, parallel light coming in from any direction is again going to be focused somewhere on the retina. So down here, if we, of course that one is going to be blocked by the iris, but if we had one that would come here, along with all the other parallel ones, it would be going through and have a focal length that would be at the retina as well. So remember, this is three-dimensionally, not just this one little beam, set of parallel lights beams coming through, but this makes it easier for us to imagine it. So obviously any light coming at the most peripheral of our vision is going to fall in the portions of the retina that have less color vision, a lower resolution, and are more monochromatic. So our peripheral vision doesn't have the highest level of acuity, but we can detect things there, right? So a lot of these things we know about. Place your fingers at the side of your head at the level of your ear and come forward. When can you begin to see them in your peripheral vision without moving your eyes? And that's all related to your finger being way over here, or over here where you can't see it, and ultimately coming into view to where some of the light coming from your finger hits the retina, and you can pick it up and begin to see that it's there. And then of course if you orient to it and focus your eye on it a little bit, then you bring the image onto retina that has higher and higher acuity. And if you look right at it and you're trying to cut your fingernails without cutting your skin and bleeding, you're using more of your fovea. Okay, so let's move from that practical way of thinking about it again to imagining a lens of a certain length and flatness, more or less flat, having a focal length that we can call the image distance, to relate it to where the object image would be projected. And if it is projected outside of the orbit itself, then that means that the image distance, or Q, is greater than the focal length of the lens, F, and the object distance is P. So we have these three factors, P, Q, and F, and they would be related to determining whether someone would have, whether you'd be able to see it in focus or not. Right. So if Q equals F, then you've got the best situation where the image distance equals the focal length, and it's actually projected onto the retina, and you can see it. So the lens would be rounded for the close vision. So objects that are closer to the lens require a rounder, a greater convexity or curvature to reduce the focal length, so that the image distance equals the focal length, and you can project it directly onto the retina. And so this is what we do when we focus our eyes, if you will. We put effort into it to bring about the changes that will relax the lens, so that it will shorten and thereby become a little bit fatter, thicker, and bend the light at a greater angle. So this is, as I stumbled through it, poorly described accommodation. So it's the process by which the eye adjusts the shape of the lens to keep the objects in focus. And I was trying to make it a little bit generic and was struggling with it, right? So we know that in the ciliary body, as we talked about, there's a ciliary muscle, so it is radial in its orientation. These are smooth muscle fibers. And the ligaments suspend the lens to the ciliary body, and so the lens is attached to the ciliary muscle by inelastic ligaments, the zonules. And so when the ciliary muscle is relaxed, and so remember, this is like a sphincter muscle, the same as the smooth muscle in the iris. So when a sphincter contracts, it's going to close down the opening that it's regulating, right? And this isn't really an opening; it's just a ring of ligaments attached to some extracellular connective tissue, right? It's highly specialized. So when the ciliary muscle is relaxed, then you're going to have the greatest diameter of this sphincter, right? It'll be the most opened. And then what that's going to do is then passively put tension on the ligaments and pull them tight and stretch and flatten the lens. And the more flattened lens is for more distant vision. So we are going to want to relax or turn off the neural impulse to these for distant vision. So when the ciliary muscle is relaxed, the ligaments pull on and flatten the lens. And then when we are focusing our attention on something, or focusing our eyes on something, then we are activating the neural input to the ciliary muscle to contract it and tighten that sphincter so it's closer to the center. And what that does is that releases the tension on the ligaments and they slacken, and then the younger lens can round up for close vision. And if you are older, even though you get the ligament slackening, the lens is going to stay more like this, because connective tissue properties just kind of change with age. Things don't work the way they're supposed to. I didn't have to start wearing reading glasses until I was about 50, so somehow I was on the later side of the curve, but still I am there, and virtually everybody reaches that point. So when the ciliary muscle contracts, it releases tension on the ligaments, and the lens becomes more rounded. Now then, we can consider common visual defects. So in cross-reference, I won't be saying it, but you need to know the innervation, so which cranial nerve innervates this, and what's the circuitry to trigger the activation of the neural input here, and what is the circuitry for inhibiting it. Not necessarily specifically with all the terminology, but you do need to cross-reference that information with the cranial nerves and put it into perspective. Kind of fill in the blanks of the presentation here. So the common visual defects, the hyperopia, or farsightedness, occurs when the focal point falls behind the retina. So the retina is on the innermost portion, and we'll look at the anatomical details of that in the next series of slides here. And the sclera is the outermost portion. So if the focal point is the natural focal point for that individual, the anatomy of their eye is at more at the sclera than it is at the retina, then and this is when you have the flattened lens, then it will be out of focus for the retina itself. So that would be hyperopia. And this, of course, can be corrected by a monoconvex lens that will add a little bit to the curvature of the front side of the anatomical lens. So it begins to bend the light, to converge the light a little bit in front of the eye, so that the natural lens can complete that and put it right on the retina. Myopia is nearsightedness, so it's obviously the opposite of that. The focal point falls in front of the retina. So here you want to use a concave lens. So this is not as convex on the front side, and it is concave on the back side. So the overall curvature is more concave toward the front. And what that'll do is that'll spread or scatter the light rays a little bit before they hit the lens, and then it'll increase the focal length of the bi-lens combination there. And of course, the ophthalmologist has specialized set of lenses that are placed in front of the patient's eyes, and then they're asked, which looks best and stuff like that. So it's corrected for the exact offset in focal length and object length for the person's eye. So, not to give it away entirely with an example, but based upon these P, Q, and F, you should carry it over to this, what equals what for a correct eye, and how would they be offset for these and for the correction. So for those three variables, how would they differ for hyperopia and myopia? The electromagnetic spectrum is shown in this slide, and we know that the photoreceptor cells in the human retina is sensitive to a range of wavelengths from a low energy of a higher wavelength. So wavelength and energy are reciprocally related. So the higher the value for the length of the wavelength, 750 nanometers, for example, the lower the energy, which is the more of the red. So it's Roy, red, orange, yellow, green, blue, indigo, and violet, if you recall. So the Roy G BIV ranging from 750 to 380 nanometers. And in wavelength, and the 380 nanometers being the higher energy. Yeah, so this, sorry, doing a few lectures here, getting a little brain tired. How about you? Anyway, the lower the number for the wavelength, the higher the energy. So it is reciprocal. Sorry, my brain just got a little confused on that for a moment. Perceptual things. And we know that that is within a very small portion of the overall electromagnetic spectrum. And you know that some organisms have capabilities beyond that of humans. And if there are devices that can take, for example, the infrared and shift it into the range that our receptor cells are sensitive, then we can see during the dark, right? Okay, then the summary of the anatomy of the retina is shown in this slide. So to the left is where the light will first hit, and that would be the aqueous, the vitreous chamber with the vitreous humor in it. So this is toward the center of the orbit, and then the periphery of the orbit would be to the right. So after the vascular layer, the first layer of the retina we would see is the optic nerve layer, if you will, or it's the layer containing the central processes of the first cellular layer, the ganglion cell layer, And then these receive innervation from bipolar cells. So remember, this is one area where we said there were true bipolar neurons, special sensory neurons. And the dendrites of the bipolar neurons, of course the axons are innervating the ganglion cells. The dendrites are receiving input from the photoreceptor cells themselves, and we know that for color there's the cone, and the monochromatic is the rod. And we'll look at these in a bit more detail as we proceed. There's two other cellular populations for local integration within the retina. Those that innervate let's just say the ganglion cells, and those that innervate the bipolar cells are the amacrine cells and the horizontal cells. There's obviously much more detail to the anatomy here, and even the function of the horizontal and amacrine interneurons within the retina, and that's beyond the scope of our course. So we're going to focus attention on the anatomy and function of the two receptive cells, and then talk a little bit about the nature of signal transmission, signal transduction with the first neuron, the bipolar cells, and then consider some of the properties of the ganglion cells. And of course, along the way, we'll see this outermost layer that is the pigmented epithelial cell layer. So in this slide, even though we're using the same color spectrum, we are using it to get our orientation of one end or the other. We are not talking about any type of distribution of receptors that pick up the different colors, as you know. What this is reminding us is that the fields of view are sent to the opposite orientation of the orbit when they hit the retina. So there is a complete crossing through the center of this. And this is not just right and left fields of view. Remember, there's a superior, an upper, and a lower field of view as well if you put your three-dimensional space into perspective. So something that is light coming from the ceiling that you pick up in your peripheral vision, or if you put your hand up right in front of your eyes and then move it up without moving your eyes to track it until you can't see it, the light coming from your hand when it just comes into your upper field of view is hitting the lower retina. So that would be as if the red was your hand here, then it hits the lower retina, right? And so it's the effort to keep this three-dimensional perspective in mind is very important. But also what is important is that this is the way the information is sent to the cortex, and then the visual perception portion of it on the brain side corrects it. It knows that what's at the bottom is really in the environment at the top. And that's, of course, through neural circuitry. And that magical step of consciousness and awareness, right, that we really don't quite understand yet. We can study it and have understandings of how it works, but we don't really know exactly how perception is transduced into conscious awareness and things like that. So the rods and cones will be seen in the next series of slides. We'll take a look at the pigment epithelium layer, and we note that it has the melanin granules in it. That's to absorb the light so it doesn't get reflected and scattered, have any backscattering. You lose resolution when you're doing that. You can have better night vision if you reflect light back, but you compromise the resolution a bit. But still, for cats, if they see something moving, if they just pounce toward the center of the movement, they pretty much figure they're going to get a mouse, right? Anyway, and then we'll go through the slides and through this next series, and they'll focus on these different parts, and we'll consider it. Reminding ourselves that the light is passing through the axonal layer and the cellular layer before it gets to the deepest portion, the most peripheral portion, where the actual chemical sensory system exists. It's physical, so it's special somatic afferent, but it's still biological chemicals that are associated with detecting the photons. So we're starting most peripheral, deep to the sclera, of course, at the pigmented epithelium, and then we'll move our way centrally to the inner layers of the retina. So what we notice is that there's a very specialized relationship between the pigmented epithelial layer and the outer segments of the photoreceptor cells, the cones, obviously kind of conical shaped, and the rods, more cylindrical shaped, looking like a rod. But within each of these are discs, and the visual pigments will be in the membranes of those discs. So notice that the discs are suspended within the cytoplasm in here, and there is still a cytoplasmic membrane encasing it, so there's cytoplasm or cytosol floating around all of these structures, and they still are a subcellular compartment of the entire cell, but there's just these connecting stalks that keep them interconnected. But remember, all the energy supplies and new chemicals to keep making these and to maintain them are coming from the cellular portion that we'll see as we proceed. But before proceeding, let's look at this detailed structure for the rod, where there is a turnover of the discs, and at the apical portion of the cell, or its outermost outer segment portion, we're going to see old discs that are phagocytized by the pigmented epithelial cells. So this will just pinch off and kind of drop off that discarded portion of the cell and the cellular contents, the discs, and then that'll be endocytosed by the pigmented epithelial cells, and any raw materials that can be will be recycled. So if we then zoom in on the discs, we see that they are lipid bilayer membrane-bound structures in a disc formation. If you remember your plant anatomy, kind of like the thylakoid discs in the plastids, the chloroplasts. And you can see that there are obviously proteins within those membranes, and that's what we see here. They split it a bit, probably shouldn't have, but anyway, we can easily imagine one of these little dumbbell-shaped structures here as being an integral membrane protein. Here's the phospholipid bilayer that we should recognize, and the membrane-spanning segment of this complex protein. They're showing it kind of as the typical solid type of a structure, but really what it is this type of protein that has alpha helical portions that are the membrane spanning segments, 1, 2, 3, 4, 5, 6, 7, and then extracellular loops in between those segments, and cytosolic loops, and then the loops inside the disc, sorry. Hopefully you corrected me there. Huh? This is all intracellular. And then the membrane-spanning segments that are lipophilic, because they can tolerate being within the lipid bilayer, also hold this lipid-soluble molecule in there, retinal. So there's an affinity for this retinal within this portion of the rhodopsin molecule. And we'll see that when the light ultimately hits it, in here, it's going to displace this molecule and bleach the rhodopsin to opsin. Okay, so that's just the anatomy part of it. We'll get to that function and put it in sequence again. So now, as we come from the outer segment to the inner segment of the receptor cells, this is where we've got all the major organelles and metabolic operations, such as the synthesis of the photopigment and the ATP production from the mitochondria. So we get the DNA and the RNA, and the ER, the Golgi, the mitochondria, all to maintain all these parts of the cell, the inner segment, but as well the outer segment. So they're very busy little buggers. And then we can follow, of course, any event that would occur in the outer segment. We know that there's going to be some polarity, some charge across the membrane that will change based upon the photons impacting these structures here. The actual photon detector system will be converted to a membrane potential that will change along here and then cause some type of chemical signaling at the synapse with the bipolar cells. And then, of course, the bipolar cells will, as we saw, communicate, have synapses with the ganglion cells. Excuse me. So, before leaving this general anatomical consideration of the two types of photoreceptor cells, the cones and the rods, there's actually subpopulations of cones, as we see here, for their maximum sensitivity based upon the photopigments that are produced for different wavelengths of the spectrum that we said are sensitive to. So here's the lower wavelength, the 400 shown here, out in the violet range. So that's a higher energy level. And the higher wavelength, lower energy level down in the red range. So there's blue cones, green cones, and red cones. And as with any photopigment, they have a range of sensitivities to wavelengths with zones of the highest or peak sensitivities. And this blue set is obviously in the blue, and then the green is in the greenish yellow, and then the red there. And as you know, combinations of these three colors can produce pretty much all the others. And then the rods are monochromatic in that they are sensitive, although at a peak close to the green range, across all, and their lowest sensitivity or lack of sensitivity is in the red range. Well, now let's consider the phototransduction in the rods. Just a moment, I highlighted a few comments in the text, and I want to make sure I bring those to our attention as we're going along. Okay, so here we are considering a rod. So this is in this wavelength here, and we are going to consider what is going on in darkness, and then when light is present, and then in the recovery phase. So in darkness, the rhodopsin is inactive. So it is inactive, called rhodopsin because it's opsin that is complexed with the retinal, as we had shown in the previous illustration, and it's shown here. The retinal has the little bend in this tail on it. And the cyclic GMP levels are very high, so the transducin protein is a G protein that is keeping the enzyme activity high to take GTP and make cyclic GMP, so those levels are really high. So it's similar to the cyclic AMP, but this is the GMP, and so there's going to be a phosphodiesterase that will inactivate this, break the cyclic portion of it, and you'll just have GMP, not cyclic GMP, to inactivate it. But anyway, it's being produced and broken down at a rate where more is being produced than is being broken down, so the cyclic GMP levels are high. When those are high, then the cyclic nucleotide gated ion channel in the cytoplasmic membrane associated with the extracellular space is open, and the influx of calcium and sodium, because this is typical extracellular fluid with high sodium and high calcium, that influx will be continuous. And what that influx is doing is countering the potassium efflux, so the potassium channels are remaining open, and this generates a membrane potential in the dark of about minus 40 millivolts. So it is somewhat depolarized compared to what would be expected as a resting membrane potential of about minus 40. But in these types of cells, they're not really they're specialized neuronal receptor cells anyway. What we'll see is they're never resting, really. There's no resting membrane potential. They are either closer to 40 or closer to minus 40, or closer to minus 70, and the rate of glutamate release at the synapse with the bipolar cell will be regulated by the membrane potential. So the closer to minus 40, they're not going to fire action potentials, but it is closer to what we might think of as a threshold to activate the calcium, the voltage-gated calcium channels here, so that there is a tonic release of neurotransmitter onto the bipolar cells. So it gets kind of tricky for vision. Without light, there's transmitter release. And then, as we see, when light bleaches the rhodopsin, which now we call opsin, so it bleaches it, and the affinity for the retinal is reduced, and then the retinal is released because the affinity is low, so it unbinds and is more likely to be in the unbound state. So then it will diffuse away because it's lipid soluble, and it gets into the pigmented retinal epithelial layer, and there are enzymes there to activate the retinal. And then the remaining protein that no longer has that molecule in it is called opsin, and it's bleached pigment. And then what this will do is it will activate the transducin, and what this will do then will cause a decrease that will inhibit the enzymatic production of the cyclic GMP. So inactivation of the existing pool will continue, and then ultimately we'll get a new lower level of the cyclic GMP. And because that's lower, the CNG channel that depends upon a high level of cyclic GMP to have a large number of them stay open will will now close, and so calcium and sodium will no longer be going in. This means that now the potassium efflux will take over, and if you remember your Nernst Goldman equation and Nernst equation, the membrane potential will now approach that equilibrium potential of potassium, which was the minus 90, I think, wasn't it? So it goes to minus 70. And so the membrane hyperpolarizes, and at this hyperpolarized level, there's less of the voltage-gated calcium channels opening, so you get less exocytosis, so the amount of neurotransmitter release into the synapse with the bipolar cell will decrease in proportion to the amount of the light. So the more light, the less glutamate in the synapse. So it's really like the reverse, the opposite of what we would think. Well, the light's going to excite cells. Well, this is where I want to read you a portion from the text in case you don't have it there to look at right now, and you can look at it again later when you do have the text available. But it isn't mentioned in the slides. So the phototransduction in the rods, in this final portion, in the recovery phase, the retinal recombines with opsin. So it's converted to the inactive form. Then it can diffuse back, out of the retinal, epithelial, pigmented epithelial cells, and the opsin, without the light around, increases its affinity for it again, and then when it binds to it, then it becomes the rhodopsin. It's inactivated, and we now call it rhodopsin again. So when the light activates rhodopsin, that second messenger cascade is initiated through transducin, as we saw in the previous slide. Now, relative to that glutamate release, we need to think about what is happening to the bipolar cells. So after the glutamate's released from the photoreceptors onto the bipolar neurons, this is that first stage of the signal processing, and there can be some integration within the retina, as we talked about. But in thinking of this synapse, there's two types of bipolar cells, light on and light off, and that'll be relevant to the overall sense we want to get of how the retina detects light and turns that into a perception centrally. So the light on bipolar cells are inhibited by the glutamate release in the dark and are activated, released from inhibition in light. So we always think of glutamate as an excitatory transmitter, as it's been in every case that we've talked about so far. But here there are some types of bipolar cells, the light-on bipolar cells, that are basally inhibited by the glutamate. So when you decrease the glutamate, then they become excited. So when light is present, these light-on bipolar cells become excited because they are disinhibited. The light-off bipolar cells are, as we always think of, excited by glutamate release in the dark and inhibited in the light. So when glutamate is excitatory or inhibitory, whether it's excitatory or inhibitory, depends upon the type of glutamate receptor on the bipolar neuron. So a light-on bipolar neuron will have one type of glutamate receptor in its membrane, and the light-off bipolar will have another that's typical for excitation in that latter case, the light-off. Sorry. So in this fashion, one stimulus, light, creates two responses with a single neurotransmitter, and then the bipolar cells then form either excitatory or inhibitory synapses with the ganglion cells, the next neurons in the pathway, and the ganglion cell receptive fields are the next thing that we want to think about. So the scientists have now identified multiple types of ganglion cells in the primate retina. Because they're more superficial on the inside and accessible to scientists, they're more readily investigated. But what they've determined in the primate retina is that there's two predominant types that account for about 80% of the retinal ganglion cells. So there are large magnocellular ganglion cells, also called M cells for short, and they're more sensitive to information about movement. And then small parvocellular ganglion cells, or P cells for short, that are more sensitive to signals that pertain to the form and fine detail, like the texture of objects excuse me, in the visual field. So this becomes a very important additional feature to what we're considering here for the ganglion cells, the receptive field. So obviously the integration within the retina is complex, and we're trying to touch on just a couple of key concepts to begin our studies and knowledge base on their function. So the receptive fields are the next thing to think about. How do we resolve images in the retina through the integration? So convergence and divergence become important aspects here. So there can either be the on-center off-surround field or the off-center on-surround field. So if there's bright light, and so each ganglion cell has a circular, really well-defined circular receptive field, whereas the peripheral sensory receptors we talked about were more irregular in shape. These are highly regular and circular. So if the light is more intense just right at the center, then it excites the ganglion cell. If it is the on-center off-surround field, if it's the type of ganglion cell that has an off-center on-surround field, then when the light is focused in the center of its field, it's going to inhibit that ganglion cell. And then there's a bright light. If the bright light is on the surround, then for the on-center off-surround, it'll be off for the surround. It inhibits the ganglion cell. And for the off-center on-surround, it's going to excite it because it's on the surround. I know that gets pretty confusing, but just to hear it, but you need to think about it a bit, and maybe writing it out or illustrating it will help you. Then if there's just diffuse light that's evenly distributed across the center and the periphery or the surround, you're going to get a weak response from both of them. So the differential responses provide a set of information. So what's important to think about here, and it generalizes to neural circuitry and how it contributes to conscious awareness and perception in general, or even subconscious integration, is that the turning off of a neuron, decreasing its firing rate, can be just as important of a set of information as turning on or increasing the firing rate of a neuron. And it's the degree to which they change, being inhibited or activated, that contributes to the signal information. So the retina uses contrast rather than absolute light intensity for better detection of weak stimuli. So the visual fields and the binocular vision are shown in this slide, and I believe it is the final slide. And there's a few points that we want to make from this slide. We are reminded we have the blue and the red for the, and again we're assuming a dorsal view, so we have the right visual field is shown in the pinkish to red, and the left visual field in the light to the dark blue. So the blue is the left visual field that is going to be represented or projected to the right visual cortex. So you recall that from before. So the left visual field is projected onto the right retina, and the right retina goes to the right cortex. But there's going to be points across this visual field from the far right to the far left, the complete visual field, where the same objects are going to be impacting both right and left retina. And so both visual cortexes are going to be picking up information from the same objects in our field of view. And this can be, even for distant and close structures. So where they can overlap in that some of the information is being projected to both primary visual cortices, and you can see the intensity of the red here as representing this most centralized portion of the visual field, and it corresponds to the highest intensity blue on the contralateral visual cortex. So we have some degree of representation of both fields of view, and the degree to which that overlap is the greatest determines the amount of binocular vision that is recreated through integration in the cortex. So the more peripheral structures in our field of view will not give us a very strong perception of depth, and where they do overlap and produce that depth perception, we call it the binocular zone. And outside of that, of course, it's the monocular zone. So the cortex merges monocular information from the two eyes to give us binocular view of our surrounding information, and the information from on-off combinations of the ganglion cells is translated into sensitivity to line orientation in the simplest pathways, or into color, movement, and detailed structure in the most complex pathways. So each of these attributes of visual stimuli is processed through a separate pathway, creating a network whose complexity we're just beginning to unravel, as the authors stated. But in our sense, it's a sufficient amount of information to take into account at this level of study. I appreciate your time and attention to this, and we'll look forward to seeing you again soon. So in summary then, thought that was the last slide, my big closure, right? So we saw the general properties, the four types of sensory receptors, through this whole series of sensory physiology. We talked about adequate stimuli, threshold, receptive field, and perceptual threshold. And again, we only looked at it in certain cases, but it is true across the board. The different modalities, the localization, intensity, and duration, the somatic senses, chemoreception, the ear, and then in this one, finally, the eye.